Theme: Stratified and glandular epithelia. Plan. Number 1. Structure, localization, functions of different types of stratified epithelia. Number 2. Regeneration of epithelia. Number 3. Glandular epithelium. Stratified epithelium is divided into 3 types: keratinized, non-keratinized, transitional. In contrast to simple epithelium, stratified epithelium has 2 or more layers of cells. Because of this, it is more durable and can better protect underlying tissues. The name of the specific kind of stratified epithelium depends on the shape of the cells in the apical layer. Cells in the apical layer of this type of epithelium are flat. Those of the deep layers vary in shape from cuboidal to columnar. The basal deepest cells continually undergo cell division. As new cells grow, the cells of the basal layer are pushed upward toward the apical layer. As they move farther from the deeper layers and from their blood supply in the underlying connective tissue, they become dehydrated, shrunken, and harder, and then die. At the apical layer, after the dead cells lose their cell junctions, they are sloughed off, but they are replaced continuously as new cells emerge from the basal layer. Types of stratified epithelium. Stratified squamous epithelium can be 2 types: keratinized and non-keratinized. In keratinized stratified squamous epithelium, the apical layer and several layers deep to it are partially dehydrated and contain keratin. It is a tough, fibrous protein that helps protect the skin. The keratin fills the cells and the organelles die. Non-keratinized stratified squamous epithelium does not contain keratin in the apical layer and several layers deep to it, and remains moist. The organelles remain in this epithelium. Stratified squamous keratinized epithelium. Localization: skin epidermis. Structure: on fingers, soles, and palms it consists of 5 layers: stratum basale, stratum spinosum, stratum granulosum, stratum lucidum, and stratum corneosum. Other parts of skin do not contain stratum lucidum. Function: barrier, protective, participation in hydrosaline exchange, resist friction, and this type of epithelium is also impermeable to water. Cells of stratified squamous keratinized epithelium. In stratum basale, columnar keratinocytes are present synthesizing protein keratin and melanocytes. In stratum spinosum, multiangular keratinocytes with spikes and melanocytes. In stratum granulosum, flattened keratinocytes with keratohyalin granules are located. In stratum lucidum, flattened cells without organelles and nuclei with keratolinin granules are located. And in the stratum corneosum, dead corneal squama are. In this slide you see skin of finger stained by hematoxylin eosin. Upper part of skin is epidermis. Its epithelium is stratified squamous keratinized. Stratified squamous non-keratinized epithelium. Localization: oral cavity, esophagus, eye cornea, vagina. Structure: consists of 3 layers. Number 1, superficial layer of flat cells. The second layer, of spine-shaped cells, and lower layer, basal layer of cells. Function is protective. Cells of stratified squamous non-keratinized epithelium. In the basal layer, columnar epitheliocytes are located on basement membrane. In the middle layer, spine-shaped cells, irregular multiangular cells. In the superficial layer, cells are flat. In this slide, you can see section of eye cornea stained by hematoxylin eosin. Upper part of eye cornea is stratified squamous non-keratinized epithelium. Transitional epithelium, urothelium. Localization, urine passages, renal pelvis, ureter, urinary bladder, urethra. Here you can see transversal section of ureter, and innermost lining of the organ represents urothelium. Structure consists of 3 layers: superficial layer of cells, intermediate layer, and lower layer is basal layer of cells. Function permits distension. Cells of urothelium. Basal layer contains round, dark cambial cells, which are source of regeneration. Intermediate layer contains polygonal shaped cells. In the superficial layer, large 2 or 3 nuclear cells having pear-shaped or a flattened shape. During stretching, shape of superficial epitheliocytes are flattened. During contraction of the organ, they are pear-shaped. Regeneration of covering and lining of epithelia. The sources are stem cells able to division during whole life of organism. Usually, these are low cells located on basement membrane in the deepness of epithelium. Stem cells of stratified epithelia are located in basal layer, in pseudostratified epithelia, in basal cells. In simple epithelia, they are located in definite parts. For example, in small intestine, stem cells are located in epithelium of crypts. In stomach, they are located in epithelium of pits. In cervix, of proper glands. Glandular epithelium. Glandular epithelium consists of stroma and parenchyma. Stroma is connective tissue with vessels and nerves. Parenchyma is epithelium made up of glandular cells with marked secretory function. Classification of glands. Glands are classified into 3 types: glands of internal secretion, endocrine, glands of exocrine secretion, exocrine, and glands of mixed secretion. Endocrine glands are ductless. They secrete their products into the blood or lymphatic vessels. Exocrine glands secrete their products via ducts onto the external or internal epithelial surface. Exocrine glands are classified according to the nature of their secretion, their mode of secretion, and the number of cells, unicellular or multicellular. In this slide, you can see unicellular exocrine glands, which are goblet cells producing mucinogen. Structure of exocrine gland. Exocrine gland consists of terminal secretory section and efferent duct. Secretory section is made up of glandular cells on basement membrane, and efferent duct is lined by different types of epithelia. According to structure, exocrine glands are divided into simple and compound. In simple exocrine glands, non-branched efferent duct is, and according to shape they are tubular and alveolar. Compound exocrine glands have few branched efferent ducts, and according to shape of these ducts they can be tubular, acinar, or tubular acinar. In this slide, you see section of uterus stained by hematoxylin, and here simple tubular glands are visible. Secretaries cycle consists of 4 phases. The first phase, absorption of initial products by glandular cells. The second stage, synthesis and accumulation of secretion in them. Third stage, excretion of secretion from glandular cells. And the last stage, regeneration of their structure. During the first phase, inorganic substances, low molecular organic substances, water, pass into glandular sites from blood and lymph. The second phase is characterized that from these substances, secretion is synthesized in endoplasmic reticulum. This secretion is accumulated in Golgi complex and granules are formed. During the third phase, granules of secretion are excreted from glandular cells, and during the fourth stage, glandular cells are regenerated either by intracellular regeneration or from stem cells in holocrine type of secretion. Mechanisms of excretion of secretion. Depending on these, glands, exocrine glands can be divided into 3 types: merocrine, apocrine, and holocrine. Merocrine type of secretion. In merocrine type of secretion, glandular cells completely keep their structure, for example, cells of salivary glands. Apocrine secretion. In apocrine type of secretion, a small portion of the apical cytoplasm is released along with the secretory product. Example, cells of mammary glands. Holocrine secretion. Holocrine type of secretion is accompanied with accumulation of fat in cytoplasm and complete destruction of glandular cells. Example, cells of sebaceous glands. According to chemical composition of secretion, exocrine glands are divided into serous, when secretion contains protein, mucous, containing mucus, seramucous, both protein and mucus secretions are sebaceous, when secretion contains sebum, and salty, these are sweat glands. Thank you for attention.